uh, we will see in the last lecture there is viral replication. uh, Viral replications first step is the attachment. After the attachment there is the next step, the penetration or a entry into the host cell. Penetration is an energy dependent process. Virus may be penetrate into host by endocytosis, translocation and fusion. uh, Means a phagocytosis. What phagocytosis or endocytosis? Here we can see the whole virus particle can be penetrate into the host body. Okay. In membrane fusion, where the envelope virus like a HIV virus can be fusion of envelope protein with plasma membrane of host cell, and then nucleocapsid enters into cytoplasm, whereas a virus envelope remain attached to the host cell membrane. And in the third type, the that is the injecting nucleic acid or a translocation. Here you can see the bacteriophage cannot be penetrate uh, because of a rigid cell wall, so only nucleic acid is injecting into the host cell. Okay. Then there is a third step, that is the uncoating. Uncoating refers to removal of degradation of capsid, means uncoating, there by releasing the genome into host cell. The virus genome is transported to the site where transcription replication can be. In some, there is no degradation of capsid, as capsid protein play a role in a viral transcription and replication. By penetrating capsid enters cytoplasm with the help of a lysosomal enzyme. Then the next step is a genome replication. Here the viral genetic material or a genome is multiplied within the host. Simultaneously, viral structural protein like capsids are synthesized. Type of genetic material varies from virus to virus. With respect to this, all viruses are divided into 7 groups by Dr. David Baltimore in 1971. Dr. David Baltimore shared Nobel Prize with Renato Dulbecco, Howard Martin, Temin in 1975 for their work or interaction between tumor viruses and the genetic material of the cell. There are the 7 groups of virus replication. Before these 7 types of virus replication, we can complete our virus replication. The next step is the biosynthesis. In this step, the following viruses components are synthesized, that is the nucleic acid, capsid protein and enzyme required for various stages of virus replication. Then after the synthesizing this component, there is a assembly where viral nucleic acid and protein are packed together to form a progeny virus, means a new virus particle, that is a nucleocapsid. Then it takes place in a host cell nucleus or cytoplasm. Here we can see which virus is assembled into nucleus and which is assembled into RNA. There the DNA virus assembled into nucleus and RNA virus assembled into cytoplasm. Okay. Then after the assembly or packaging, there is a step of maturation. It takes place in either host cell nucleus or cytoplasm membrane. Then the after the maturation, there is a release of daughter virions occurs either by lysis of host cell, by non-enveloped virus and bacteriophages, or budding through the host cell membrane, that is the enveloped viruses. During budding, they acquire a part of the host cell membrane to form the lipid part of their envelope. In this way, the viruses are replicate. Now let's we see the 7 group of Baltimore classification, which are categorized on the basis of genome or a genetic material of viruses which contain. Here the 7 types are: first is a double stranded DNA, second is a single stranded DNA, third is a double stranded RNA, fourth single stranded positive sense RNA, fifth single stranded negative sense RNA, sixth single stranded positive sense RNA with DNA intermediate, seventh double stranded DNA with RNA intermediate. Now let me start with the first, the double stranded DNA. Here the double stranded DNA can be converted into double stranded DNA with the help of cellular or viral DNA polymerase, or it may be go to form mRNA with the help of cellular RNA polymerase 2. This mRNA synthesizes a protein, and this double stranded DNA, it is a nucleic acid for the double stranded DNA virus. Here, after the synthesizing the protein, it will assemble or form a virus structure. That is, here you can see the replication of genome of double stranded DNA virus. There the example are pox virus and herp virus. Now we will discuss the second type, single stranded DNA. Here you can see the single stranded DNA which undergoes the cellular DNA, with the help of it will form a double stranded DNA. There is a cellular DNA polymerase enzyme works. After the formation of double stranded DNA, it will be formed mRNA with the help of RNA polymerase 2. This mRNA form a protein, and this double stranded DNA which are converted into single stranded DNA. This single stranded DNA is a genetic material, and the formation or a production of this protein can be assembled with together and form a single stranded DNA virus. There are the example Pircovirus and Parvovirus. Now see the double stranded RNA. Third type is a double stranded RNA. Here the double stranded RNA which is undergo the viral RDRP to form a positive sense single stranded RNA. This positive sense single stranded RNA are work as a mRNA and form a protein component. And after this there is a formation of double stranded RNA, which are the main genomic material of this cell of this virus. After the protein synthesis and the genetic material are packed together and to form a double stranded RNA virus. Here the examples are reovirus and orbibus. Then the fourth type is a single stranded positive sense RNA. Single stranded positive sense RNA work as a mRNA, and it's directly undergo to form a protein. It synthesizes a protein. Then the viral RDRP enzyme is used to convert the single stranded RNA to negative strand single stranded RNA. Then negative single stranded RNA will convert into positive single stranded RNA with the help of viral RDRP. After this conversion, the nucleic acid, single stranded positive sense RNA, and the protein which are compactly bind together and form a single stranded positive sense RNA. Here you can see the replication of this genome of positive sense single stranded RNA virus. There is the example Toga virus and hepatitis E virus. Now there is a fifth type of single stranded negative sense RNA. single stranded negative sense RNA, uh, which may be which are